Welcome back to our second episode of Faith and Science in the News. This is Frank Lyon, your host. At the end of the last podcast, you were introduced to Dr. Antony Flew. He was, as we mentioned, the most famous atheistic philosopher of last century. Several years before his death in 2010, he was forced by the evidence from science to change his mind concerning his worldview, becoming a theist, a believer in God. He describes the initial public announcement of his newfound worldview in his book, There Is a God. In 2004, Dr. Flew was at a symposium in New York that also included Israeli scientist Gerald Schroeder and Scottish philosopher John Haldane. He was asked if recent work in the origin of life pointed to the activity of a creative intelligence. This was his response, and I quote, "Yes, I now think it does, almost entirely because of the DNA investigations. What I think the DNA material has done is that it has shown, by the almost unbelievable complexity of the arrangements which are needed to produce life, that intelligence must have been involved in getting these extraordinarily diverse elements to work together. It's the enormous complexity of the number of elements and the enormous subtlety of the ways they work together. The meeting of these two parts at the right time, by chance, is simply minute. It is all a matter of enormous complexity by which the results were achieved, which looked to me like the work of intelligence." Close quote. What had just been going on prior to this pronouncement by Dr. Flew? What had he been observing? It was the Human Genome Project that began in 1990 and declared complete in April of 2003. It took 13 years and about $1 billion to sequence the human genome using many labs around the world, led by the U.S. National Institutes of Health, NIH. You've been hearing a lot about NIH recently related to the coronavirus. Let's ask a question to get started here. What is a genome? The NIH website defines a genome as an organism's complete set of DNA, including all of its genes. Each genome contains all the information needed to build and maintain that organism. In humans, a copy of that entire genome, more than 3 billion, and that's with a B, DNA base pairs, is contained in all cells in your body that have a nucleus. Let's unpack all this terminology. What was it about DNA that impressed Antony Flew? What is DNA? Where is it found? Before we get started with this conversation, let me mention that there is a blog associated with this podcast. Please comment and/or ask any questions via this blog. Also, I will use the blog to provide you links to other resources. For today's topic, I have provided some links to animations to help you better understand the molecules and the processes that we will be discussing. You will find the link to the blogs at the top of the webpage. The animations will help you to understand some pretty complicated processes we're going to be talking about. We'll try to make them pretty simple. But I would suggest you listening to the podcast, then check out the animations, and then come back and give the podcast another review and kind of get a feeling for what we're talking about here as we discuss, in generic terms, what's called protein synthesis. Back to DNA. The acronym stands for the name of a large macromolecule called deoxyribonucleic acid. It is the information-carrying genetic material. Part of the genome is comprised of genes, each of which code for the manufacture of a protein molecule. Some of the code does not make up genes, but is information that is used to regulate many biochemical processes. DNA is also generically known as a nucleic acid. It is one of the 4 major molecules found in living organisms. Besides nucleic acids, these 3 others are proteins, lipids, which we call fats, and carbohydrates, the sugars. So we have these 4 different molecules that biochemistry is all about: nucleic acids, proteins, lipids, and carbohydrates. Now, the structure and composition of DNA was not determined until the 1950s. Before that time, we really had no idea how information was passed between generations of cells or organisms. The structure of DNA initially first identified by Francis Crick and James Watson, working from X-ray diffraction analysis that had been provided to them by Raymond Gosling, working in the lab of Rosalind Franklin. What they discovered was that 2 very long polynucleotide chains were coiled around each other to form a double helix. There were 3 component molecules making up DNA, 3 parts: a phosphate group, a 5-carbon sugar, and a nitrogen-containing base. A good way to visualize the DNA molecule is as a ladder. The rails of the ladder are the 2 polymer chains made up by alternating phosphate groups with the 5-carbon sugar, known as deoxyribose. This descriptive name is due to a missing oxygen atom when compared to the sugar ribose. The rungs of the ladder are made up of one of 4 different nitrogen bases. The bases are joined together by hydrogen bonding at the center of the rungs of the ladder. The 4 bases can be classified into 2 groups, one group known as the pyrimidines and the other group known as purines. In DNA, there are 2 pyrimidines, cytosine and thymine, and 2 purines, adenine and guanine. When describing the genetic code along the DNA molecule, these bases are commonly abbreviated by their first letter: C, T, A, and G. And interestingly, the A will only form a bond with T, and C only with G. Now, imagine taking a hold of the rails at both ends of the ladder and twisting them in opposite directions, top and bottom, forming what has been described as a double helix. The hydrogen bonding between the bases that make up the rungs of the ladder down the center of the axis of the DNA acts as a zipper that can be unzipped and rezipped by breaking and then reforming the hydrogen bonds among the 4 bases: cytosine, thymine, adenine, and guanine. Again, I just remind you, check out the links in the blog to get a better visual idea of how the molecule is put together. There's some pretty good animations there I have left links to for you to look at on the internet. Now, another question: where do we find DNA in the cell? Living organisms can be divided into 2 major groups based on where we find the DNA located: prokaryotes and eukaryotes. Eukaryotes, which include all plants and animals, have cells that contain membrane-bound organelles, one of these being the nucleus. The nucleus contains the DNA wound tightly into chromosomes, 46 chromosomes in the human. In contrast, the prokaryotes, bacteria and blue-green algae, do not contain membrane organelles. The DNA is literally naked within the cell. Let's kind of change the focus here. We'll change the attention here to discuss how the information in the DNA molecule is used, and we'll focus specifically on these eukaryotic organisms. Again, that includes human beings. As we mentioned, the Human Genome Project was able to demonstrate that our DNA is made up of 3 billion base pairs. We have described how each of these pairs make up a rung in a very long, twisted ladder. Most cells in the human body, about 40 trillion by recent estimates, contain a copy of the DNA molecule. On the average, a human cell divides every 24 hours. At each division, the cell must undergo a process called replication, where the DNA unzips, reproduces a copy of itself, zips back up, and using a process called mitosis, one copy goes into each of two new daughter cells. How does the message along the DNA lead to the manufacture of protein molecules? We have it stored here in the DNA. How do we get that information and synthesize, manufacture these protein molecules by this process commonly called protein synthesis? To discuss this topic, we need to introduce another type of nucleic acid to our discussion. It is called ribonucleic acid, or RNA for short. It also contains a 5-carbon sugar, in this case ribose, that has that extra oxygen atom compared to the deoxyribose sugar found in DNA. It's not a helical structure, but a linear piece of information. And indeed, with these nitrogen bases, thiamine, there's a little change here. It contains a different base called uracil. And now, instead of thymine binding to adenine, as in DNA, you have uracil binding to adenine. One type of RNA is messenger, or mRNA. It copies the genetic code from the DNA molecule using a process called transcription. Messenger RNA then takes that copied code through a pore in the membrane surrounding the nucleus out into the cytoplasm of the cell to find an organelle called a ribosome. The ribosome is made up itself of about 50 different proteins. The ribosome is where proteins are manufactured. Now, proteins are a chain of another chemical. They're called amino acids. The amino acids must be in a very precise order for them to fold and function properly as protein molecules. There are only 20 amino acids used to make up all the proteins in the body. Out in the cell, we find each amino acid bound to a single molecule of another RNA, known as transfer RNA or tRNA. The message laid down by the 4 nitrogen bases in mRNA, in that particular molecule, are read in groups of 3. This 3-letter sequence is known as a codon. As an example, let's take the codon A, G, U. Remember, A, adenine, G, guanine, U, uracil. Uracil instead of thymine in the RNA, as we discussed. Now, the transfer RNA has the opposite code, the anticodon, programmed into it, which would be UCA. The transfer RNA, with that anticodon, binds only to a single type of amino acid, serine. This process is called translation. As the code is read, 3 letters at a time, it is translated into the proper sequence of amino acids to make a functioning protein molecule as the individual amino acids are bound together in sequence. At present, it is thought that about 1 to 2% of the human genome is divided up into individual genes that code for proteins. There appear to be at least 20,000 to 25,000 genes, ranging in size from a few hundred base pairs of these nitrogen bases to more than 2 million base pairs. This is the complexity of specified information that Dr. Anthony Flew was referencing. The complexity becomes even more astounding when we consider that many proteins in the form of enzymes, molecular machines, are required for the process of transcription and translation to occur. So when transcription occurs, where messenger RNA is taking that code from the DNA molecule, be able to transfer it out to the ribosome, there's several protein molecules involved in that process. Here's just a short little list. RNA polymerase, sigma factor, repressor and activator proteins, exonucleases, endonucleases, spliceosomes, and we could go on and on. numerous proteins involved in that process of manipulating those nucleic acids. Now, the same is true for the translation process. As I mentioned a little bit earlier, that ribosome itself is made up of 50 different protein molecules all working together to manufacture a protein. And there are many more molecular machines required in this manufacturing process. Now, a point I'd like to make here to think about for just a minute, based on what I just said. Think about this. The production of proteins requires these nucleic acids, DNA and RNA. But the production and the manipulation of DNA and RNA requires proteins. Scientists now know that translation actually requires more than 100 different proteins. That's led some biologists, Richard Lewontin being one, to ask what makes the proteins that are necessary to make the protein. David Goodsell states, the unanswered question in biochemistry is this: which came first, proteins or protein synthesis? We have here the classic situation of which came first, the chicken or the egg. I hope you have not got bogged down in the details. The take-home message is that the cells in our body are full of genetic information that is highly specified, precisely ordered, and results in very specific functions related to the molecular biological processes going on in our bodies. Just like words in a book, except this code will fill many books. Bill Gates stated that DNA is like a computer program, but far, far more advanced than any software ever created. Does all this look like the product of unguided, random variation? Or does it strike you as the kind of information that has to have a mind behind it? I'm reminded of David writing in Psalms chapter 139, verses 13 to 14. You, and he's addressing God here, you created my inmost being. You knit me together in my mother's womb. I praise you because I am fearfully and wonderfully made. And we see an example of that as we look at the human genome. How probable is it that this whole mechanism of protein synthesis just came together by chance, not requiring a supernatural designer? Antony Flew decided it was not possible. We will further answer that question in subsequent podcast after defining some terms our next time together. Terms that will include science, evolution, Darwinian evolution, microevolution, macroevolution, and we'll mention theistic evolution. Be sure to check out the blog for additional resources. We look forward to your feedback there. Thanks again for joining us here at Faith and Science in the News. Also, thanks again to the Blues to Joy Ministry for providing the platform to make this podcast available. Until next time, it has been my privilege, Frank Lyon, to be your host.